This afternoon, we're here with Matt Handel, the CEO of ExpressCells. And Matt, you got your start in the life sciences space working with big pharma. And it sounds like you found so exciting on the entrepreneurial side that you made a shift. Tell us a little bit about your background and how you got to where you are today.
Oh, thanks. It's a bit of a wander. It's a bit of a journey, actually. I'm a big pharma refugee. Went to business school up at MIT. Afterwards, was looking at options. One of them, for example, would be marketing in a paper products company, and I knew I wanted to do something more meaningful. I ended up joining Merck, which was a great company to join in the 90s. Actually spent some time as a sales rep out in California. And I truly believe everyone should spend time in sales because you can't understand the customer unless you're meeting with the customer. But I had a pretty traditional path forward at Merck, market analysis, product marketing, worked on some great products like Agristat and Singular. Eventually left Merck, and after a brief stop in medical education, joined Shire Pharmaceuticals. And again, joining Shire Pharmaceuticals in 2004 is just being like put in the cockpit of a supersonic plane. It was growing rapidly. It was a great place to be. Ran business analysis and forecasting a couple of times on a global basis, ran a product group, but it got me into business development, and that was amazing. A lot of fun to do deals, but the other part, it was a lot of fun to be on the other side of the table from small companies that were doing incredible things. And it really made me, come to the recognition I should be on that other side of the table. These were small groups. They were fast moving. They were really, on the cutting edge of science, and it was great to interact with them. So in 2016, I jumped to the other side. This is now my third startup, and this is the one that has made the most progress.
So tell us about that pivotal moment when you did that, got started with this company. I think you had been working closely with scientists at Temple, and he had some ideas. Tell us about that.
Yeah, well, my partner is Dr. Oscar Perez Leal. He's a professor in the Temple School of Pharmacy and the Mulder Center for Drug Discovery. We had started one company together. We had actually been introduced by the Science Center, and that was a therapeutics company. As with many, we ran into some technical issues. But one day, Oscar and we were at a reception. He pulled me into a side room to start showing me these pictures of cells just glowing. He said that these were all genetically edited cells, that he had developed a new technique to do this. They had filed a patent, and there was a big business opportunity. And I said, I have to think about it for a bit. And it took a few months, but by then he had made progress. He was in a Temple Accelerator program, came on board. We incorporated less than 2 years ago, and it's just been a rocket ship again ever since. We've been moving incredibly quickly.
Well, that's incredibly exciting. So tell us what you actually do. You've got a really unique spot that you're developing these cells, and so let's hear more about that.
Sure. So we create cell lines for research. For people who need a little more explanation, cell lines are cells that are grown outside of an organism. So they're cultured in a laboratory. Very often the ones we work with are immortalized, which means that they keep growing and growing. What we do are called knock-ins. We insert genes into cells. And if people ask, why would you ever want to do that? There are two examples. One is we create what are called tags. We can take genes that code for fluorescent proteins, the ones that you see in jellyfish or eels, for example, and attach them to other genes in the cells, which means that they light up protein. So if you want to see the nucleus of the cell, we can make it glow green. If you want to see the mitochondria, we've done that and made them glow blue. Or if you've got a special protein you're targeting, say one that you think that if you inhibit, you can treat a form of cancer, we can tag that for you as a custom project. The other thing we can do is overexpress proteins. So take that same protein, the one that you think is a cancer-causing agent, we can set it up that cell lines will produce more of it, which makes it easier to target and easier for a researcher to study.
So you mentioned earlier that your process is a technical sale process, and that often you're really customizing to the needs of your clients. Tell us a little bit more about that in terms of how you find new clients and why clients are coming to you, the specific capabilities and the factors that differentiate your company.
Sure. Well, let's talk about two types of clients, as you said. There are the ones who know they need a knock and sell line. We're not the only people doing it. We do believe that we have a much more advanced process, one that better fits the customer needs. So when people are looking, they might say, I need a knock-in. I need to tag a particular protein with a green fluorescent protein. You can find us online. We're very active in terms of our promotion online. And that's when they know what they want and they can come to us. But there are times when we want to work with customers as well. So we have 3 manufacturers reps we've engaged. And so they can work with a customer who may not start with, I need a knock and sell line, but who are saying, I have a problem, or I'm working on a specific experiment. And in those cases, they can work with them to have a conversation about why our cell lines could help them save time and money.
So we talked a little bit earlier about your funding cycle. You've received Series A funding. You've built the company around that. And now what's next?
Well, 2 areas. One, get revenue. The best funding is the one where a customer pays you and you can make a profit off of that. I'm not saying we're profitable now, but that's our goal. The second is we're looking to do an additional raise, probably because the lawyers would be angry. I can't get into all the details, but we're looking to raise money and it'll let us do 3 things. One, it'll let us expand our operations, hire scientists, do some capital expenditure, allow us to have more throughput, and maybe speed up our process as well. The second, it allows us to do more promotion. And number 3, product development. It's always good if you can take the type of work we're doing and add more products to the offering. If you think about it, we currently have a catalog right now of 8 products that are available for anyone who wants to buy them. We'd like to be able to, significantly increase that.
So you're currently at Biolabs at 3675 Market Street.
Mm-hmm.
Why have you focused on Philadelphia? We're excited that you're here. Why is Philadelphia the place for you to be?
Because we're a Philadelphia technology. The technology comes out of Temple University. The tech transfer, did require us to take both the L and the subway to get here, but that's better than transferring it, thousands or hundreds of miles away. I also think it's because we have a great talent pool here in Philadelphia. Our lab director, Shamim Nagdi, is just fantastic. She was at Jefferson for 10 years. She's the example of why we want to be in Philadelphia, to find people who really have a great skill set and the willingness to, apply it in an entrepreneurial way.
Matt, oh, go ahead.
I should also add, I know what it costs for a lab space up in Boston. Boy, I like paying less.
Yeah, that's great. Well, we really appreciate your time today. Your company, ExpressCells, provides a service and materials that are so integral to the drug development process. And it's really good to get some insight into that. We wish you great luck in your financing, and we look forward to checking in with you down the road as you continue to grow the company. So thanks very much for being with us today.
And thank you for having me.